The human eye is functioning as a spectrometer, analyzing the light reflected from the surface of a solid or passing through a liquid. Although we see sunlight, or white light, as uniform or homogeneous in color, it is actually composed of a broad range of radiation wavelengths in the ultraviolet, visible, and infrared portions of the spectrum. The component colors of the visible portion can be separated by passing sunlight through a prism, which acts to bend the light in differing degrees according to wavelength. The colors are red, orange, yellow, green, blue, indigo, and violet. Electromagnetic radiation, such as visible light, is commonly treated as a wave phenomenon, characterized by a wavelength or frequency. Visible wavelengths cover a range from approximately 400 to 800 nanometer. The longest visible wavelength is red and the shortest is violet.
Beer-Lambert law. The method is most often used in a quantitative way to determine concentrations of an absorbing species in solution, which is A equals to log 10 times I naught over I, which is equals to epsilon times C times L. A is the absorbance. I naught represents the energy before it passes through the sample, and I is the energy after it passes through the sample. L is the path length through the sample, and lastly, C is the concentration of the absorbing species. For each species and wavelength, epsilon is a constant known as the molar absorptivity or extinction coefficient. The Beer-Lambert law is useful for characterizing many compounds, but does not hold as a universal relationship for the concentration and absorption for all the substances. Absorption spectroscopy carried out in this region, which is visible or near ultraviolet, are sometimes called electronic spectroscopy. The diagram drawn is the various kinds of electronic excitation that may occur in organic molecules. Of the 6 transitions outlined, only the 2 lowest energy ones, which is the leftmost, ticked in red color, are achieved by the energies available in the 200 to 800 nanometer spectrum. As a rule, energetically favored electron promotion will be from the highest occupied molecular orbital to the lowest unoccupied molecular orbital, and the resulting species is called an excited state. When sample molecules are exposed to light, having an energy that matches a possible electronic transition within the molecule, some of the light energy will be absorbed as the electron is promoted to a higher energy orbital. An optical spectrometer records the wavelength at which absorption occurs, together with the degree of absorption at each wavelength.
The instrument used in ultraviolet visible spectroscopy is called a UV BIS spectrophotometer. The basic parts of a spectrophotometer are a light source, mirrors, filter, cuvette, monochromator, or a prism to separate different wavelengths of light, detector, and a computer for display purpose. A beam of light from a visible and UV light source is reflected by a mirror and is filtered before passing through the monochromator. The function of monochromator is to separate and transmit a narrow portion of light as the input of process. The scanning monochromator moves the diffraction grating to step through each wavelength so that its intensity may be measured as a function of wavelength. When the light reaches the beam splitter, it is split into 2 beams before it reaches the sample. One beam is used as the reference, and the other beam passes through the sample. The intensities of these light beams are then measured by electronic detector. The intensity of the reference beam, which should have little or no light absorption, is defined as I0, while the intensity of the sample beam is defined as I. The transmittance can then be calculated using the ratio I over I0, and it is usually expressed as a percentage. The absorbance can then be calculated using the value of transmittance by using the formula A equals to negative log T. The ultraviolet region scan is normally from 200 to 400 nanometer, and the visible portion is from 400 to 800 nanometer. The data obtained is then displayed in graphical form for further analysis.
Ultraviolet and visible spectrometers have been in general use for the last 35 years, and over this period have become the most important analytical instrument in a modern-day laboratory. In many applications, other techniques could be employed, but none rival UV visible spectrometry for its simplicity, versatility, speed, accuracy, and cost-effectiveness.